Good morning, dear students. Yesterday, I told you about the process of reproduction. Reproduction in the living organisms takes place by two mechanisms, that is, asexual reproduction and sexual reproduction. Asexual reproduction involves only one parent, while sexual reproduction involves two parents. There are different modes, there are different ways in which an organism can reproduce by asexual mode of reproduction. Yesterday we discussed about them. Today we are going to recapitulate what we discussed yesterday. First mode of reproduction by asexual mode of reproduction is by fission. Fission stands when an organism breaks down itself to give rise to new organisms. So the organism itself loses its individual entity while the new organisms are produced. It can take place by binary fission, like we discussed about amoeba and leishmania. And it may also take place by multiple fission, like it takes place in the plasmodium. Plasmodium is the causative agent of malaria. It can reproduce by multiple fission, in which the cell of plasmodium can divide itself into many new cells. Another mode of asexual reproduction is by spore formation. Spore formation takes place in rhizopus and other fungi, in which they produce spores, and each spore can give rise to new fungi. Another mode of asexual reproduction is by fragmentation. We discussed fragmentation in Spirogyra, in which a filament of Spirogyra can break down and each new piece can then give rise to new filament. Next mode of asexual reproduction that we discussed was regeneration. I told you about regeneration in planaria, in which accidentally, if planaria breaks down into pieces, each piece can give rise to new planaria. Next mode of reproduction is by budding. In this case, a bud appears on the surface of the parent organism, and this bud then grows and detaches itself from the parent and gives rise to a new individual. The next is vegetative propagation. Vegetative propagation is when a new plant can be grown from its vegetative parts, like its roots, stems, or leaves. Next is tissue culture, in which a tissue, a group of cells, is cultured in vitro. In vitro stands for in lab conditions. So in lab conditions, we are growing a tissue from the plant, which can give rise to a new embryo, and then it can be transferred to a test tube where it will give rise to a new plantlet. This new plantlet can then be grown in nurseries and then transferred to fields. This is a mode of reproduction which is mainly used for the research and scientific purposes. That's all for the recapitulation. I hope I am clear with the topics. If you have any doubts, please feel free to contact me. Thank you. God bless you.